Immunology Insights, Basics and Beyond, Chapter 61, Georges Köhler and the Development of Monoclonal Antibodies. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our recent chapters, we have looked at monoclonal antibodies, checkpoint inhibitors, and adoptive cell therapies, powerful tools that all rely at some point on having a single, well-defined antibody with a precise specificity. Today, we go back to the moment when that became technically possible. Our focus is Georges Köhler, co-inventor of the hybridoma technique and one of the key architects of the monoclonal antibody era. So let's talk science. Who was Georges Köhler? Georges Jean Franz Köhler was born in Munich in 1946 and studied biology in Freiburg. Early on, he became fascinated by one of immunology's central puzzles: how the immune system can generate an almost unlimited diversity of antibodies. In the early 1970s, he joined César Milstein's lab at the MRC Laboratory of Molecular Biology in Cambridge. At that time, everyone knew that individual B cells make highly specific antibodies, but nobody knew how to capture a single B cell clone and make it produce exactly the same antibody forever. Serum from an immunized animal was always a polyclonal mixture, useful but messy and irreproducible. Köhler and Milstein set out to solve exactly that problem. Now let's come to the hybridoma breakthrough. The core idea of the hybridoma method is elegant. Fuse an antibody-producing B cell with an immortal myeloma cell to create a hybridoma that is both immortal and monoclonal. In practice, the process involved 4 key steps. First, immunization. Mice were immunized with a defined antigen so that their spleens contain B cells producing antibodies against it. Second, cell fusion. Spleen cells were fused with myeloma cells using a fusion agents such as polyethylene glycol. A few of these fusions produced viable hybrid cells. Third, selection in het medium. The fused cells were grown in het medium, which kills unfused myeloma cells, which lack a key enzyme, and unfused B cells, which are not immortal. Only the hybrids survive. In that context, het stands for hypoxanthine, aminopterin, and thymidine and is a selection medium used in the hybridoma method. Aminopterin blocks de novo DNA synthesis, so only cells that can use the salvage pathway, thanks to the enzyme from the normal B cells, survive by using hypoxanthine and thymidine. Unfused melanoma cells lack these enzymes and die. Unfused B cells are not immortal and die as well, leaving only the fused hybridoma cells that produce the monoclonal antibody. Fourth, cloning and screening. Surviving hybridomas were cloned and each clone was tested for secretion of the desired antibody. The right clone could then be expanded indefinitely. The result was a stable cell line producing one antibody with one specificity in virtually unlimited amounts. For the first time, immunologists had access to reproducible, precisely defined antibody reagents. Let's take a moment to see how they worked at the time within the lab. Immunize mice, fuse spleen cells with myeloma cells, plate them out, and test hundreds of wells for antibody production, mostly to be rewarded with negative results or unstable cultures that fizzled out. Then came a batch of plates where some wells kept producing the same antibody over and over again, passage after passage. At first, it looked like just another set of screenings. No fireworks, no dramatic eureka. But as they checked and rechecked the data, it slowly sank in. These hybrid cells had inherited immortality from the myeloma partner and specificity from a single B cell. People who worked with Köhler recall that he reacted not with grand gestures, but with a dry, almost amused satisfaction. The big breakthrough felt to him like what science often really is. A lot of quiet, repetitive work until, almost casually, one of the plates on your bench turns out to have changed the world. In keeping with the culture at the MRC laboratory of molecular biology at the time, neither Köhler nor Milstein filed a patent on the hybridoma method. The technique was effectively placed in the public domain, which allowed laboratories and companies worldwide to adopt it rapidly and build entire industries on top of it. Financially, this meant the inventors never saw more than their academic salaries, but scientifically, it turned out to be one of the most profitable non-patents in modern medicine. An idea so valuable that it changed the toolbox of every immunology lab on the planet almost overnight. For Köhler, the intellectual satisfaction of cracking how to make a single defined antibody line mattered far more than any missed royalties from patents. An example of how curiosity-driven basic science, not financial planning, often lays the foundation for the most impactful medical breakthroughs. Now, let's look closer at why monoclonal antibodies changed everything. The impact of hybridomas was enormous. First, for basic research, monoclonal antibodies made it possible to define and track individual cells and molecules with unprecedented precision. The CD, or cluster of differentiation system, for immune cell markers, for example CD4, CD8, CD20 and so on, emerged directly from this technology. Second, for diagnostics, monoclonal antibodies became central to ELISAs, rapid tests and immunohistochemistry, enabling reliable detection of hormones, infections, autoantibodies and tumor markers in clinical laboratories. And third, for therapy. The hybridoma concept laid the foundation for therapeutic antibodies such as rituximab, trastuzumab and many others. Even though today's drugs are often chimeric, humanized or fully human, they all trace their origin back to Köhler and Milstein's ability to isolate that very first monoclonal clone. In 1984, Georges Köhler, César Milstein and Niels Jörn received the Nobel Prize in Physiology or Medicine for this work, a rare case where the methodological innovation reshaped an entire field. Now let's assess the way from mouse hybridomas to modern drugs. The first monoclonal antibodies were fully murine, and when given to patients, they often triggered human anti-mouse antibody or HAMA responses that limited efficacy and caused side effects. This drove the next wave of innovation. First, chimeric antibodies, murine variable regions fused to human constant regions, reducing the immunogenicity. Second, humanized antibodies, only the murine antigen binding loops, so CDRs, retained on an otherwise human framework, even further reducing immunogenicity. And then third, fully human antibodies, generated using phage display or transgenic mice carrying human immunoglobulin genes, completely avoiding immunogenicity. All these refinements kept the key advantage of monoclonals, precise specificity, while making them better tolerated and longer lasting in humans. None of this would have been possible without the original hybridoma method that allowed individual antibody specificities to be captured in the first place. To conclude, after returning to Germany, Köhler became director of the Max Planck Institute of Immunobiology in Freiburg, continuing his work on antibody diversity and B-cell biology. His life was tragically short. He died in 1994 at only 48 years of age. But his scientific legacy is woven into almost every corner of modern immunology. Every time we stain cells for CD markers by flow cytometry, run an ELISA, or treat a patient with a monoclonal antibody or antibody drug conjugate, we are quietly relying on the technology he helped create. His story is a powerful reminder that basic methodological research, done carefully, patiently, can ultimately change how we diagnose and treat disease on a global scale. In our next chapter, we will stay in the world of immunotherapy, but move beyond cancer, exploring how similar principles can be used to calm down harmful immune responses in immunotherapy for autoimmune diseases. Before closing, I have a small request. If you enjoyed this episode and want to hear more, Please like and subscribe. It's a small step for you, but it helps me a lot. So thank you for listening and see you next time.